Hiya, so my name is Mary Moore. I'm a PhD student in exercise physiology, and I'm in Dr. Scott Rector's lab. And today I'm going to talk a little bit about my dissertation project that I've focused on over the past couple of years. So my dissertation focused on the role of hepatic mitochondrial function and mitochondrial turnover in the development and progression of non-alcoholic fatty liver disease, and also lifestyle modification to treat this particular disease. And so just a little bit of background. NAFLD, or non-alcoholic fatty liver disease, is a spectrum of liver disease that ranges from a buildup of fat in the liver called steatosis, which can then progress with inflammation to non-alcoholic steatohepatitis, or called NASH. And then if NASH is allowed to progress or is left untreated, it can then progress with fibrosis to cirrhosis or end-stage liver disease. And end-stage liver disease ultimately can result in pathway to liver transplantation. The reason why NAFLD or NASH is an important disease to study is because it's becoming the leading cause of liver transplants in the developed world. And it's currently also, there's no pharmacological treatment available in order to treat NAFLD. And so our lab, multiple mechanisms are thought to contribute to the development and progression of NAFLD. And one of these factors is hepatic mitochondrial dysfunction. And this is what our lab focuses on. While hepatic mitochondrial dysfunction and its contribution to NAFLD or NASH progression or development is well characterized in animal models, it's poorly understood in humans. And so what my dissertation mainly focused on was looking at the phenotype of mitochondrial dysfunction across the spectrum of NAFLD in a human population. So in the first part of my dissertation, we looked at how a loss of mitochondrial function, mainly fatty acid beta oxidation and electron transport chain efficiency, and also how the mitochondria deal with oxidative stress and how this contributes to human NAFLD. In the second part of my dissertation, we looked at how changes in mitochondrial turnover can contribute to worsening NAFLD NASH phenotype in humans. And mitochondrial turnover is characterized by three main factors, and this is mitochondrial biogenesis, which is the production of new healthy mitochondria, mitochondrial dynamics, and mitochondrial autophagy or mitophagy, which allows for the breakdown of old dysfunctional mitochondria in order to replenish the healthy mitochondrial pool in the cell. So mitochondrial function and mitochondrial turnover have been well characterized in animal models in terms of their link to NAFLD and NASH, but we know little about this in humans. And what my dissertation showed was when we looked at mitochondrial function and quality control in a human population, we showed that there's a decrease in ex vivo fatty acid oxidation, and this was associated with increased or no change in electron transport chain efficiency. But it's also associated with increasing oxidative stress, particularly with worsening NAFLD phenotype. And this was associated with poor mitochondrial morphology, in that the mitochondria, when we looked at them under the microscope, they tended to look more swollen and hypodense, which is associated with worsening mitochondrial function. And we also showed that with this worsening mitochondrial function, there was a reduction in the ability of the cell to produce new healthy mitochondria via biogenesis. And there was also a reduction in the ability of the cell to promote the breakdown of old dysfunctional mitochondria. So once we phenotyped this in a human population, we then looked at how we could potentially treat or reverse NAFLD or NASH in the human setting. And so the mechanisms, two main factors that we focused on was weight loss via diet and exercise. And so it's well characterized that weight loss via diet and exercise can histologically reverse NAFLD and NASH, but the mechanisms by which this occurs remains poorly understood. What we showed is when we underwent a 9-month supervised lifestyle intervention in which we recruited patients to an active treatment group and had them lose weight via caloric restriction and also via supervised exercise routine for 9 months, we showed that NAFLD, this type of lifestyle treatment was associated with a reduction in histological NAFLD outcomes. This was highly correlated with greater reductions in fat mass in response to the lifestyle treatment, and also increases in aerobic capacity in response to the exercise treatment. And we also showed that there were significant reductions in steatosis and ballooning. And that's the main crux of what my dissertation focused on. And thanks for listening.